the separation of mixtures into individual components.
Chromatography means color writing. With this technique, the components of a mixture in a liquid medium are separated.
The separation takes place in absorption and capillary.
The paper holds the substances by absorption. Capillary pulls the substance up the paper at different rates.
Pigments are separated on the paper and show up as color streaks.
The pattern of separated components on the paper is called a chromatogram.
And this will be what our project is about.
The chromatography process for each one of the different plants, and this will help us to take out the pigments of them.
The problem of our experiment was how can we separate the different pigments of the leaves.
If we use a chromatography process, then we can separate the pigments of the plants because of the molecular weight of different pigments. Materials we will use for our experiment are safety goggles, chromatography solvent, chromatography paper, ethyl alcohol, and fresh spinach.
Test tube rack, scissors and ruler, fresh leaves of plants, and glass stirring bowl.
Paper clip, cork, mortar and pestle, sand, and 10 milligrams of graduated cylinder.
Is the spinach. The experimental group are the other two plants that we choose.
Dependent variable is the type of leaves. The dependent variable is the different pigments observed in the chromatograph. The constants, the same solvent, the sun environment, the same filter paper, and the same time left.
Experiments, we noticed all the pigments because of the chromatograph that we took out. We found out that the spinach, our control group, had different pigment colors, like yellow, which is xanthophylls, blue-green, which is chlorophyll B, and yellow-green, which is chlorophyll A. All of these have different RF values. For example, xanthophyll had 5 over 8.5 that give us 0.58. Chlorophyll B had 2.6 over 8.5 that give us 0.3, and chlorophyll A had 1.5 over 8.5 that give us 0.17.
With our second sample, we weren't so lucky like with the spinach. We only got two pigments extracted, xanthophyll and chlorophyll. The RF value of xanthophyll was 5.3 over 8.5. That is 0.62, and the RF value of the chlorophyll was over 8.5, that is 0.35.
The third sample showed us three pigments as well as the first, which were yellow meaning xanthophylls, blue-green chlorophyll B, and green-yellow meaning chlorophyll A. The RF value of the xanthophyll was 6.2 over 8.8. That is 0.71. The RF value of the chlorophyll was 5.5 over 8.7, that is 0.6. The RF value of the chlorophyll was 3.2 over 8.7, that is 0.36.
We think that this experiment wasn't very accurate as well as others that we have done. We did separate the pigments of the plants, but some of them weren't very well extracted because of the plants that we brought. We couldn't see them very clearly. Our hypothesis actually was supported and it was useful. The only thing that was kind of wrong in our experiment was that one of the plants that we brought was very difficult to smash, so we couldn't extract that much of the liquid unlike the other two. That actually affected us when we wanted to extract the pigments. We think that for further experiments, the teacher needs to tell the students what type of leaves they need to bring so they could extract the pigments really well, and it will work as they wanted to.